Hello and welcome to the present lecture on the topic of derivatives of integument. The integument refers to the outermost protective covering of the animal body, that is the skin, and its various derivatives. The skin itself is relatively very simple, but its derivatives are numerous and they are very complex. Depending upon the layer from which they are derived, the derivatives of the integument fall into two categories, that is epidermal derivatives and dermal derivatives. Epidermal are derived from epidermis and dermal derivatives come from dermis. Epidermal derivatives, as you can see, they comprise of epidermal glands, scales and scutes, digital cornifications, horns, feathers and hair. Whereas dermal derivatives includes dermal scales and scutes, that can be dermal scales of fishes, dermal scales and scutes of tetrapoda, and dermal fin rays of fishes. First we will be taking epidermal derivatives, and the first one that we will be discussing is epidermal glands. The glands of the integument are epidermal glands. They are formed by the Malpighian layer of the epidermis. They often invade the dermis. They may be unicellular or multicellular. They may be simple tubular, alveolar, compound tubular, compound alveolar, or compound tubulo-alveolar, as you can see in the slide here. These glands are lined by cuboidal cells or columnar epithelium. These are usually named after the nature of their function. The 9 major different types of epidermal glands are as mucus glands, poison glands, luminescent glands, femoral glands, uropygial glands, sweat glands, sebaceous, scent, mammary glands. Mucus glands, as the term mucus indicates, they are responsible for the production of mucus. These glands actually secrete mucin, which forms slimy or sticky mucus on coming in contact with water. This mucus has the role of keeping skin moist and slippery, and it protects against the harmful bacteria and fungi. This is abundant in amphibian skin. However, it is found in the skin of cyclostomes, fishes, etc. as well. This moist skin helps in respiration of these organisms as well. Poison glands are also the cutaneous glands. They secrete poison. The poison may be bitter, irritating, and even dangerous to the predators. These modified multicellular cutaneous glands, they are larger, but they are fewer than mucus glands. For example, the poison glands of the amphibians, in case of some amphibians, they collectively form a mass called parotid glands. Now, the luminescent glands are the light-emitting organs. They are known as photophores. In deep-sea luminous teleost fishes, certain multicellular epidermal glands form these photophores. You can see in the slide here as there are photocytes, and the photocytes are covered over by the mucus cells. They form the magnifying lens, and there is the reflector layer at the base of these photocytes. The reflector is made up of guanine crystals. The light emitting may not be continuous. This is flashed on and off by the sympathetic nerves. The chemicals like luciferin, in the presence of the enzyme like luciferase, oxidize this oxyluciferin, and this oxyluciferin is luminescent in nature. It produces light. This luminescence is an adaptation in dark, deep oceans, and it is the means to communicate with the mates or the surroundings. Now the femoral glands, they are found in male lizard, uromastyx, on the ventral surface of each thigh. They are in a single row, 12 to 18 femoral pores from knee to cloacal aperture. The sticky secretions from these apertures on coming in contact with air, it results in the production of spines, and these spines help the males to hold the females during copulation. Next is uropygial gland. It is the characteristic of birds. It is found near uropygium. It is branched and alveolar and exudes an oily secretion that is used for lubricating beak. It is used for preening feathers, attracting the opposite sex during breeding season due to its odoriferous nature. The oil secreted by it contains promatum. The promatum, which is picked up by the beak and used for preening and waterproofing of the feathers. Now the sweat glands. Sweat glands are sudoriferous glands. They are abundant in the skin of most mammals. They are coiled tubes embedded deep in the dermis, and the openings, they can directly open on the surface of the skin, or they can open into the hair root. The directly opening on the skin surface, they are called acrine sweat glands, whereas the others that open into the hair follicles, they are called apocrine sweat glands. Elimination of sweat helps in cooling through evaporation. It is also responsible for the excretion of certain urea and some salts. Sweat glands are not universally present in the mammals. They may be absent in some cases, whereas in other cases they show a restricted distribution. The colors can also vary. For example, it is a red colored sweat in case of male giant kangaroo and hippopotamus. They may occur only on the soles of feet of cats and mice. That is, they may show restricted distribution in some cases. And now the next gland that we'll be taking is sebaceous gland. These sebaceous glands are branched alveolar glands. They open into the hair follicles of mammals. In some cases, they may open directly onto the skin surface. They secrete an oily secretion that's called sebum, that is greasy in nature. This sebum keeps the skin and hair soft, greasy, waterproof, and glistening. Sebaceous glands may also be absent in some cases, and these sebaceous glands are modified to form ceruminous glands of external ear, or in another case, for example, meibomian glands of eyelids. They are modified sebaceous glands. Meibomian glands spread oily secretion over the exposed surface of eyeball. Now the scent glands, as the term scent indicates, they are the secretions of these glands are odoriferous in nature, and they serve to attract or repel mates and these foes. As you can see in the slide, they may occur on different parts of the body. In a zoo or in any cowshed, the smell there may not be due to the unhygienic conditions, but it may be due to the scent of the scent glands of these mammals or animals. Now the last major gland, that is mammary gland. This is a characteristic of mammals, and it is compound tubular gland that produces milk during lactation period for feeding the young ones. Mammary glands show teats on the surface of the body, but in some cases these teats or nipples are absent, for example in monotremes. The mammals that possess nipples, they are modified sebaceous glands, whereas the nippleless or teatless, they are the modified sweat glands. The teats or nipples, they may be true or false. The true teats contain all the gland openings at or near the surface of the teat, whereas in false teats, all ducts empty into one bag-like cistern from which a single tube leads to the tip of the nipple. True teats are found in man and apes, whereas the false teats, we can see them in these ungulates. These were the major types of epidermal glands. Now we will discuss epidermal scales and scutes. As you all know, all the hard horny structures that develop by the accumulation of a protein that is known as keratin. The keratinized or cornified, this stratum corneum, the accumulation of keratin in that leads to the production of the hard exoskeletal structures. All stratum corneum cells are cornified and they form hard, horny exoskeletal structures like scales, beak, horns, claws, nails, hoofs, feathers, hair, etc. in different vertebrates. Scales, as we see, they are many small, flat and hard pieces of keratin covering the skin of an animal, particularly of fishes and reptiles. Whereas the scutes, they are horny, chitinous, or hard external bony plates like structures. They are thick and much hard plate like scales. Crocodiles and turtles, as we see, they have large, thick, rectangular scutes. They are not overlapping, but they are touching each other, and they are supported beneath by dermal bones. They are supported beneath on below. They are supported below by the dermal bones. These scales and scutes are responsible for the protection as well as prevention of the water loss. As we see in the slide, these scales, they are found in reptiles, they can be found in birds and even in mammals. The figure here shows the scales of the green lizard. These scales and scutes, they are either molted individually or they are molted together. That is the process that's called ecdysis. The scales show overlapping arrangement, for example, the scales of the snakes. In case of birds, the scales can be present on the lower leg, the foot, or at the base of the beak. In case of mammals, these scales can be on the feet and tails of rats and beavers. There can be large scales on the body of scaly anteater, for example, armadillo. The slide here shows the thick, hard scutes on the surface of the crocodile body. The slide here shows the crocodiles and alligators have large scales, that scutes, which are shed individually. Now the next derivative of the integument, that is digital cornifications. These digital cornifications, they are the modifications of stratum corneum at the tips of digits. At the tips of digits of either hands or feet. They are as claws, they are nails, and they are hoofs. Claws, as you can see in the figure, the claws are found in reptiles, birds, and mammals. And these claws are identical in structure. It is made up of a hard, pointed, narrow, curved, horny dermal plate that's called unguis, and a less hard ventral plate that's called subunguis. And these both enclosing the tip of the digit, covering the last tapering phalanx. Nails, you can see, they are the characteristic of primates only. In case of nails, like the claws, the dorsal plate is unguis, but the unguis here is broad and flat, whereas subunguis is softer and much reduced. The tip of the digit below the nail, it is highly sensitive, and it forms a highly vascular pad that is sensitive to touch. Now the hoofs, these hoofs are characteristics of ungulate or hoofed mammals only. Unlike claws and nails, the unguis here is U or V shaped. The subunguis is also U shaped. It is greatly thickened and it is touching the ground. That is the lower side of the foot that contains subunguis. The horseshoe that can be nailed into the subunguis, and near this subunguis there is a horny substance of a softer horny substance that is called cuneus. Besides, there are other modifications of the stratum corneum like horns, feathers, hair, etc. If we see horns are found in hoofed mammals, that is Artiodactyla and Perissodactyla only. They are the organs of offense and defense. They are found at the head, and there are 5 different types of horns. Some can be true horns and some are false horns. True horns means these are the products of stratum corneum. First, let's see true horns. True are hollow horns. They occur in the both sexes of goat, sheep, cattle. They are unbranched, they are permanent in nature, they are cylindrical, and they continue to grow throughout life. They are made up of hollow dermal bony core that arises from the frontal bone of the skull, and they are covered over by an epidermal horny hollow cap or covering. Second is prong horns. The prongs means branches. The horns of prong horned antelope, that is Antelope capra. They are also true horns. It is formed by a small central permanent bony core arising from the frontal bone and covered over by a thin, hollow and horny epidermal horn. But the horny sheath of the prong horn bears 1 to 3 branches, that is, they are called prongs, and these branches are shed every year. The permanent horny, this permanent bony core, and that becomes the base around which next year a new horn develops. Now the third one is antlers. They are characteristic of the deer family only. They are found mostly in males only, but in some cases they can be found in both sexes like reindeer and caribou. These antlers are the annual growths, and they are not the true horns. It is a branching solid outgrowth of dense connective tissue connected basally to the frontal bone of the skull. Deposition of calcium salts actually make this antler hard. Growth. This antler may be covered with a vascular skin or velvet. When growth is complete, this velvet wears off, thus exposing the naked horny antler. After the breeding season is over, the antlers are shed and new antlers develop the following year. Now the giraffe horns of giraffe are stunted. These are unbranched. They are also permanent in nature, but they are antlers. Antlers means they are not shed. They are not shed annually. They are not branched in nature. They are smaller in size, and they are present in both sexes. Now hair horns, these are the fibrous horns. They are found in rhinoceros. They are found on the top of the nasal bone. There can be 1 or 2 depending upon the species of this rhino. Indian rhino has single horn, whereas African species has 2 horns, one after the another. And these horns are entirely made up of thick, horny and keratinized epidermal fibers fused together. These are permanent in nature, that they are not shed annually. Now the feathers. Feathers are characteristics of birds only. They are also epidermal in nature, and they are dry, they are non-living, they are cornified products of stratum corneum epidermis. These unique structures they are light in weight. They are a flight adaptation, as they are light in weight, and they conserve body heat. They make the surface of the body of the bird streamline, as well as waterproof. Presence of different colored pigments make them beautiful. There are different types of feathers that can be tail feathers, there are flight feathers, semi-plumes, phyloplumes, bristles, or down feathers. These were the epidermal derivatives. Now we will be discussing dermal derivatives, and this includes dermal scales and scutes. First we will see dermal scales of fishes. Scales in fishes, they are dermal in origin. These scales are covered by overlying epidermis, but it wears off as these scales develop. 5 different types of these dermal scales are known depending upon their structure or nature. For example, cycloid scales, ctenoid scales, placoid, ganoid, cosmoid. The cycloid scales, these are thin, oval, or round. These are overlapping and flexible scales, and they are found in most of the modern teleosts. Ctenoid scale, as you can see in the slide, they resemble cycloids, but they have comb-like teeth on the overlapping edges. For example, in sunfish. Placoid scales, they are characteristic of cartilaginous fishes, and they are spiny tooth-like projections on the surface of the body. Tooth-like, teeth-like projections. Ganoid scales are like placoid scales, but the substance, the animal-like substance that ganoid is found, so they're called ganoid scales. Cosmoid scales, they are placoid-like with two basal layer of bone substance, that is dentine-like cosmine, and an outer vitrodentine is found in them. Now the dermal scales and scutes of tetrapoda. These dermal scales are scutes. For example, in crocodiles and alligators, we see they have many oval bony plates embedded in the dermis, which are dermal in origin, and they are found below the scutes. They are called osteoderms. In mammals, bony plates occur in armadillos and whales. Similarly, dermal fin rays, as you can see in the slide, the supporting structures of the fins of the fishes, they are dermal fin rays. These are long flexible fin rays embedded in the dermis. For example, in case of chondrichthyes or the cartilaginous fishes, they are horny hair-like, made up of fibrous connective tissue, and so they are called ceratotricha. Cerato means horn and tricho means hair. In osteichthyes, that is bony fishes, they are branched and they are made up of series of segments or scales, and so are called lepidotricha. Lepido means scales. The series of segments or scales make them, so they are called lepidotricha. Similarly, unsegmented, sharp and spine-like fin rays, they are called actinotricha. This was all about the derivatives of the integument as per the syllabus.
T-H-E-N-K-Y-O-U. Thank you. Jazakallah.